Welcome to Should Have Known It, where we present 15-minute videos of things you definitely should have known. Hi, welcome to Shoulda Known It. I'm Ms. Aoki, and today's topic is the volume knob on your genes. Here is the version of genetics most of us carry around. You're born with a set of genes. Those genes determine your traits, and short of a mutation or a cosmic ray, that's that. Your DNA is your destiny, locked and immutable. The way a book's text doesn't change when you read it in a different room. It's a clean story, it's also incomplete. Every cell in your body carries essentially the same genome. The neurons in your prefrontal cortex and the beta cells in your pancreas have the same DNA sequence, but they behave nothing like each other, make nothing alike, respond to the world in completely different ways. Something above the sequence, epi from the Greek, meaning above, is doing the organizing. That's epigenetics. The question isn't what your genome says. The question is which parts of it are being read right now, in which cells, under what circumstances. And the answer to that question turns out to be remarkably plastic, shaped by what you eat, how often you move, how much chronic stress you carry, and what your parents experienced before you were conceived. The main molecular mechanism is methylation, a small chemical tag, a methyl group, just one carbon and three hydrogens, attaches to a cytosine base in the DNA, typically where a cytosine sits next to a guanine in a sequence called a CPG site. When enough methyl groups cluster around a gene's promoter region, they block the transcription machinery from reading that gene. The gene is still there, it's just been told to be quiet. The enzymes that add these tags are called DNA methyltransferases, DNMTs. The enzymes that remove them are called TET enzymes. Together, they perform a kind of ongoing editorial process, adding and erasing marks in response to chemical signals from the cell's environment. This isn't damage, it's regulation. The genome has a layer of annotation on top of it, and that annotation is the part that responds to life. Methylation isn't the only mechanism. Histones, the protein spools that DNA wraps around, can be acetylated, phosphorylated, ubiquitinated. Each modification loosens or tightens the spool, making the wound-up DNA more or less accessible to be read. And there are small RNA molecules, microRNAs, that can bind to messenger RNA and prevent translation even after a gene has been successfully transcribed. The system has redundancy. Multiple layers of volume control sitting above the sequence. Here is what makes this genuinely strange. These marks can be copied when cells divide. The DNMT1 enzyme specifically recognizes hemimethylated DNA, a newly replicated strand that hasn't been marked yet, and copies the methylation pattern from the old strand to the new one. So a silenced gene in a liver cell stays silenced through thousands of rounds of cell division. The annotation is heritable at the cellular level. That's not metaphor. That's mechanism. In the late 1990s, Randy Jirtle and Robert Waterland at Duke University ran an experiment with agouti mice. The agouti gene, when fully expressed, produces yellow fur and also drives the mouse to overeat, becoming obese and prone to diabetes. These mice are fat and yellow and sick. But the gene's activity is controlled by methylation at its promoter. If that promoter is silenced, the mouse is brown, lean, and healthy. Same gene, completely different animal. Jirtle and Waterland fed one group of pregnant agouti mice a diet supplemented with methyl donors, folic acid, vitamin B12, choline, betaine. These are the raw materials the body uses to build methyl groups. The offspring of the supplemented mothers were predominantly brown and lean. The offspring of the unsupplemented mothers were yellow and obese, same genetic background. Same agouti mutation. The difference was what the mother ate while pregnant, and whether that silenced the gene in the developing embryo. This result was striking because it showed that diet could shift the epigenome of the next generation in a specific, measurable way. But it's also worth holding carefully. Mice are not humans. The agouti gene doesn't have a direct human equivalent. And the diet intervention was extreme, far more targeted methyl supplementation than anyone eats. The mechanism is real. The extrapolation requires caution. The most rigorous human data comes from a natural experiment that no one would have chosen. In the winter of 1944 to 1945, a Nazi blockade cut off food supplies to the Western Netherlands. For about 5 months, civilians survived on less than 500 calories a day. Then liberation came in May 1945, and the famine ended abruptly. The cohort of children conceived or born during that period, the Hunger Winter cohort, has been studied for decades. Researchers found that people who were in utero during the famine, specifically during the first trimester, had lower methylation of the IGF2 gene 6 decades later compared to their siblings who were conceived outside the famine window. IGF-2, insulin-like growth factor 2, is involved in fetal growth and metabolism. The methylation difference was detectable in blood samples taken when these individuals were in their 50s and 60s. An environmental event that lasted 5 months in 1944 left a molecular trace that was still readable in 2008. The health consequences for this cohort included elevated rates of obesity, cardiovascular disease, and schizophrenia, though the causal pathways from altered IGF-2 methylation to any specific outcome are still being untangled. What the Dutch data gives us is not a clean mechanism story. It gives us evidence that early gestational environment can produce lasting, measurable epigenetic changes with population-level health consequences. That's a different and more careful claim than famine reprograms your genes. But it's still remarkable. Cortisol, the primary stress hormone, doesn't just make you feel tense. It binds to glucocorticoid receptors throughout the body, and those receptor complexes enter the nucleus and directly influence gene transcription. In animal models, early life stress reliably produces lasting hypermethylation of the glucocorticoid receptor gene in the hippocampus, which means fewer receptors, which means a blunted feedback loop, which means the stress response stays on too long after it's triggered. Michael Meany's lab at McGill documented this carefully in Rats, comparing pups raised by attentive mothers versus neglectful ones. Exercise has the inverse kind of story. A 2012 study out of the Karolinska Institute, Melaine Lindholm's group, had sedentary adults cycle on ergometers 3 times a week for 3 months and took muscle biopsies before and after. They found methylation changes at hundreds of genomic loci, including at promoters of genes involved in energy metabolism and muscle adaptation. Some changes appeared within hours of a single bout of exercise. The epigenome of muscle tissue is responsive in a timescale of hours to days. Tobacco smoke, certain pesticides, bisphenol A. These have all been associated with epigenetic alterations in exposed individuals and in some animal studies in their offspring. The mechanisms differ. Some chemicals directly inhibit DNMT activity, others introduce oxidative stress that disrupts TET enzymes. The common thread is that the regulatory layer above the genome is chemically accessible to the environment in ways the sequence itself mostly isn't. What this adds up to is not quite the simple lifestyle rewires your DNA headline, but it's not nothing either. Your epigenome is in continuous dialogue with your circumstances. The conversation is real. The outcomes of that conversation have health consequences. The degree to which you can deliberately steer those consequences remains, in most cases, genuinely unclear. Now we arrive at the part where pop science tends to overreach, and where the actual science is doing something subtle and interesting. The question is whether epigenetic marks acquired during one person's lifetime can be transmitted to their children or grandchildren, not through behavior but molecularly through the sperm or egg. This is sometimes called transgenerational epigenetic inheritance, and the evidence for it in humans is considerably weaker than it is in nematodes or certain plant species. Here's why it's complicated. During fertilization and early embryonic development, the genome undergoes a nearly complete epigenetic reprogramming. Methylation marks are globally erased and then reestablished from scratch, guided by the embryo's own developmental program. This reprogramming is thought to be one of biology's ways of preventing an individual's acquired experiences from being imposed on the next generation. The erasure is not perfect. Some imprinted genes, which are epigenetically marked in a parent of origin specific way, escape reprogramming, but the general rule is erasure. Yet there are studies, and the Dutch cohort is one of them, suggesting that second generation effects exist. The children and grandchildren of the famine cohort show elevated rates of certain metabolic conditions. In animal models, particularly Omer Nilsson's work on plants and Michael Skinner's work on rats exposed to the fungicide vinclozolin, epigenetic changes have propagated through 3 or 4 generations. How? The honest answer is we don't fully know. Either the reprogramming is leakier than we thought in specific contexts, or there are RNA-based mechanisms in sperm that carry information the DNA sequence doesn't, or there are indirect behavioral pathways that mimic molecular transmission. All 3 hypotheses have active research programs. It's worth naming the distortions explicitly because they're everywhere. The wellness industry version says your thoughts and feelings are rewriting your DNA in real time. And therefore, positive thinking has a molecular basis, and you can epigenetically optimize yourself. This takes a real finding. The epigenome is responsive to environment and launders it into a claim about conscious intention that the data does not support. The political overselling version says epigenetics validates the inheritance of acquired characteristics, which means Lamarck was right and Darwin was wrong, and therefore fill in your preferred ideology here. This collapses a specific, mechanistically distinct phenomenon, epigenetic inheritance in particular contexts, with a broad alternative theory of evolution that has been extensively falsified. Epigenetics doesn't rehabilitate Lamarck. It's a regulatory layer operating on top of a genome that still evolves by natural selection. The most common distortion is simply scale. A mouse study showing that a specific dietary intervention alters methylation at a specific locus gets translated as scientists discover that eating more vegetables reprograms your children's DNA. Every word in that sentence is a distortion of the original result. The intervention was not vegetables. The effect was not reprogramming. The transmission was in a controlled rodent model under conditions that don't map to human nutrition. Keeping track of those translations is not pedantry. It's the minimum requirement for thinking clearly about a field that genuinely matters. Let me be precise about what the evidence supports, because it is substantial even without the embellishments. DNA methylation and histone modification are real, biochemically well-characterized mechanisms that regulate gene expression. They respond to nutritional inputs, particularly folate and other methyl donors during critical developmental windows. Early gestational environment in humans, famine being the clearest case, produces lasting detectable changes in the epigenome with measurable population-level health effects. Exercise produces epigenetic changes in muscle tissue on short timescales. Chronic stress alters the hypothalamic pituitary adrenal axis through epigenetic regulation of glucocorticoid receptors, and this has been documented in both animal models and human populations. Transgenerational inheritance in humans is suggested by epidemiological data, but the molecular mechanism of transmission remains unresolved. The reprogramming bottleneck at fertilization is real and substantially limits what marks can be transmitted. The field is active, the evidence is accumulating, and the honest position is that inheritance of environmentally acquired epigenetic marks is plausible in specific contexts and not yet established as a general principle in humans. What that means practically, and this is the part worth carrying out of the room, is that the developmental window matters enormously. The periconceptional period, the first trimester, the first years of life, these are when the epigenome is most plastic and when environmental inputs have the most durable effects. Maternal nutrition, early childhood stress, exposure to endocrine disrupting chemicals during development. These are not just pediatric concerns, they are multi-generational public health questions, and the epigenetics research gives us a molecular frame for understanding why. Go back to the score metaphor. Your genome is a piece of music with roughly 20,000 genes, protein coding genes anyway, plus a vast amount of regulatory sequence we're still characterizing. The epigenome is the performance instructions written above the staff. This section fortissimo, that section tacit, this theme gets a repeat, that coda is cut. The musicians are your cells. They all have the same score, but the instructions they've been given and the instructions they'll give their descendants vary by location, by developmental history, by what the orchestra has been through. What you cannot do is choose your score. What you do have, partially conditionally, more so at some life stages than others, is some influence over the performance instructions. Not through intention and not through thinking positively at your cells, through the physical and chemical and social environment your cells inhabit over time. Diet during pregnancy, stress in early childhood, exercise across a lifetime, toxin exposure, or the absence of it. These are the inputs the system was built to integrate. The system has been integrating them quietly the entire time. That's not a magic story. It's more interesting than that. It means the conversation between a genome and its environment is not a metaphor. It has molecular machinery, it runs on enzymes, it produces records that can be read and compared and studied. We are at the cellular level continuously in dialogue with our circumstances. And some of what we say in that dialogue, in certain windows, under certain conditions, echoes forward. That's worth understanding clearly. The genome is fixed, its expression isn't, and the mechanisms that make that true are some of the more beautiful pieces of molecular machinery life has produced, worth knowing, worth being precise about. That's where we leave it today. Read Nessa Carey's The Epigenetics Revolution. If you want the full account, it holds the complexity without flinching from it. I'll see you next time. And that brings us to the end. Thank you for joining me and learning something new. Until next time, click like and subscribe.